Okay, so thank you, Antonella, for the introduction and Lucy's fantastic overview. So, are all protein hydrolysates the same? So the work I'm going to talk to you about has been funded through a project funded by Nestle, and it has involved people working in my team at the University of Surrey, collaborators in a mass spectrometry company called Waters Corporation in Manchester, as well as the Nestle team themselves. So this is my declarations, and I guess my biggest conflict, other than the fact this work was funded by Nestle, is my shares in Reactor Healthcare, which is a startup providing food challenges for diagnosis of food allergy. So Lucy's already gone over some of this, so I'm going to repeat it and try not to spend too much time doing it. But we had this amazing systematic review done by Carmen Rigioni and others a year or so ago about clinically relevant allergens that are important in diagnosis. And I'm looking, where's the laser pointer there? Is that working? Yeah. And I think this is really important, showing us actually, what are the important allergens in cow's milk allergy. And we obviously have the caseins here, which are very abundant and are an important allergen with 93% specificity and 67% specificity. But you also have the whey proteins, alpha-lactalbumin and beta-lactoglobulin, coming on behind, also very important allergens. So they're the focus of what we were analysing. So caseins are amazing proteins. They're the major component in milk, and what they have is a very flexible structure. They are what we call mobile, so they're constantly moving around. So there's a kind of term called rheomorphic that gets applied to them. But what's very clever about these molecules is they have phosphate groups on them. And those phosphate groups are designed to bind calcium. And the evidence is that this calcium, the protein binds the calcium and it forms what we call nanoclusters. And that is crucial because without that, you would not have the calcium content of milk that we have. This is above the solubility limit of calcium. So those of you who live in a hard water area, you will know your kettle gets caked up on the inside. That's what would happen if you just had the calcium there without the protein there. So the protein plays a really important role in this, and those nanoclusters are actually all stabilized in these very big micelles that are actually really quite large. And that's the reason why milk is white, is because those big micellar structures scatter light. And if you precipitate the caseins, which you do when you make cheese, the liquid goes clearer. So the other two players in this are our whey protein allergens, so beta-lactoglobulin, so that's 2% of milk protein. It's what's called a lipocalin, and actually other allergens you find that are lipocalins are inhalant allergens in things like animal dander. It's incredibly tough, very resistant to heat, and it's resistant to digestion. The other whey protein is alpha-lactalbumin. This is part of a very big family of proteins that are actually often involved in protection, and that includes lysozyme, which we secrete, for example, in our tears. It can bind calcium as well, but is more labile to heat and digestion. So these proteins carry epitopes, sites. An epitope is really the IgE binding site or an antibody binding site on a protein. And we have two different kinds. So the first one are the linear or continuous epitopes. And this is where you have a stretch of amino acids that the antibody binds to. And it's a whole series in the sequence, they're contiguous. But we have another kind, and in actual fact, in reality, the most of our epitopes are likely to be what we call conformational or discontinuous. And this is where different parts of the protein structure are brought together in its three-dimensional architecture. And what's important here is that conformational epitopes can be destroyed by unfolding your protein, by heating it. And that unfolding process, if you think about an egg and you boil an egg, the white goes from being a translucent, gloopy material to something that is hard and white and dense, and that is a reflection of that unfolding of that protein. But our linear or continuous epitopes are a bit more difficult to get rid of. So food processing can remove those IgE epitopes by causing that thermal unfolding, thermally induced unfolding of the protein, or you might have things like pH that actually also causes protein unfolding. But hydrolysis is another means that will change a protein structure, and this is really important because that can actually remove those continuous linear epitopes by chopping them up into smaller pieces. So, how are hydrolysates manufactured? And this is the key parts that steps that are involved in that manufacturing process. Often cow's milk infant formula can start, sometimes it can start from whole milk, but it might also start from a fraction such as whey or casein. And we've got two types of infant formula manufactured. So we've got a partially hydrolyzed protein, which most people say it's less than 5 kilodaltons, and the extensively hydrolyzed ones, where they're generally less than 3 kilodaltons. And a crucial thing that got implemented in the manufacturing 20 or more, and I'm looking at Anne here because she'll know, because she knows about this sort of stuff. At least 20, 25 years ago, we started using ultrafiltration to remove big bits of protein, and that was a really important thing, quite tricky to do and to do well. So that's our process, and the sorts of enzymes that often get used here in food industry are actually microbial enzymes, which have a broad specificity. So partial hydrolysis, and the problem with getting your hydrolysis to work with your enzymes properly is if it only does it partly, it won't remove the epitope. So this is some work that was done by the team in EAACI with Hervé Bernard many years ago, looking at an enzyme called plasmin that degraded casein, but resulted in very large fragments that retain their IgE binding capacity, either by immunoblotting or in an immunoassay. And similarly, here's another example of IgE binding capacity being retained. So this is actually increasing degrees of hydrolysis as we go this way. Don't worry too much about the DX. They were doing things with and without dextran. But the point here is to see how the IgE binding capacity reduced but plateaued depending on the degree of hydrolysis. And you can see here, this is the degree of hydrolysis that you've got here, but even this one, which is the most hydrolyzed, still got IgE binding, and even though there is much less intact protein. So getting your hydrolysis to work effectively is really important. So this is the work that Sophie Newton and the team in Nestle did some years ago now, 2020 it was published, showing if you've got a library of extensively, or products that have the words extensively hydrolyzed on them, then actually there's a huge variation in the amount of, so the gold shows the beta-lactoglobulin, the pink shows the casein, and the blue bars show the degree of hydrolysis. So as your degree of hydrolysis increased and you've got smaller peptides, you've got less retained IgE reactive or reactive BLG or casein. But there are some outliers there, but it nevertheless shows that they were not all the same. So the rationale for the project that we've done is to actually characterize the EHFs for structures that could cause an IgE-mediated reaction. So obviously residual intact protein, peptides that are at least 1300 to 1600 Daltons. So this is enough length of a peptide to accommodate an IgE antibody binding site. And also whether those peptides could aggregate, and they can make themselves into bigger structures through the formation of intermolecular disulfide bonds, or other constituents like the calcium, because if you remember, the casein binds the calcium in nanoclusters, so the calcium can help bring things together. And this is the team of people who've done the work between the University of Surrey, Waters Corporation and Nestlé. So we've used several different methods. So one method we used is SDS-PAGE, which allows us to see the proteins, molecular weight of 3,500 to 200,000 Daltons. We also use something called gel permeation chromatography, which allows us to look at aggregates, which could go up to 75,000. And we use mass spectrometry to really look at the peptides, and particularly larger peptides. And what we did in our sample preparation, we put things through a filter to keep all the big stuff, get rid of the small stuff. So this is the SDS-PAGE analysis, and we have a collection of 8 different types of EHF, and what I will say is I have no idea what these are. The work has all been done completely blinded, and that's how we're planning to publish it, but an unblinding key will be provided, because I think that is, we all will agree that's useful. So in this track, you can see this is our partially hydrolyzed formula. Lots of protein here, that's what's shown here with that smeary appearance, even up to more than 100,000. So no wonder that's no good for somebody, an infant with cow's milk allergy. But when we look here, going to EHF8, so EHF8 has hardly anything, but look at 1. I mean, it looks an awful lot like the PHF, and even 2 and 3 have an awful lot of fuzzy material here. So that's indicating that they are not the same, and some contain very large aggregated fragments. So Nestlé also did their analysis exactly as before, with looking for casein and BLG, as well as the number of peptides. And what you see is here, yeah, PHF, lots of big things, lots of BLG, casein, but then we have this gradation, and EHF 1, 2, and 3 really are not in the same place as 4 to 8. Clearly got large fragments present. So our mass spectrometry analysis that we did, I don't want you to worry too much about what we've done here, but it's called principal components analysis for those who are interested. And what it shows is that the pattern of peptides we got for EHF1 really does resemble what you see in a partially hydrolyzed formula. EHF2 and 3 are in a different place, and then all the others are clustered together. What makes the difference? Well, they could be different starting materials because we didn't know what it was. The enzymes they use in their processing might be different, and they may have other process parameters that are different across the manufacturing process. So we've actually mapped all the milk proteins. I'm just showing you here the analysis for the beta-lactoglobulin because that's obviously an allergen of interest. And what you can see here is, if we look again at the number of peptides, the total number of peptides, and these are the number of peptides over 3,000 Daltons. So this is our EHF1, got a lot of peptides, but actually had the most number over 3,000 Daltons. But if we go down, you can see 2 and 3. There are still some large peptides in EHF 4 to 7, and 8 has hardly any peptides in it at all. So that's just showing all of these have the same term on the label, EHF, so they are not the same. And this is an example of one of our very long peptides that we sequenced in our mass spectrometry, has a molecular weight of 4,322, and it contains an IgE epitope shown there in the kind of pinky orange. And the levels of this peptide varied. So we don't actually see it in the PHF, and that's because the PHF actually has intact protein, not so much peptide. But you can see here it's quite high in EHF1, it's high in EHF2, reduced perhaps in 3 and 4, slightly higher in 5. These are different lots of the same EHF, 6, but then when we get to 7 and we get to 8, we have hardly any of it. So it's showing you the power of the mass spectrometry to actually map those IgE epitopes. And I'm hoping at some point we will also be able to say, what we found with the mass spectrometry, does that bear out in practice? Have we got IgE binding capacity that reduces the clinical reactivity of that EHF8 compared to EHF1? Right, so are all protein hydrolysates the same? The answer, of course, is no. And it may well be that the heterogeneity we've seen in this study explains some of the clinical observations that some EHF can cause symptoms, and it really does emphasize that some products carrying a description EHF are actually perhaps more like a PHF, which isn't suitable for cow's milk allergy. So as I said, next step, confirm what we've done, physicochemical analysis using effector cells, and validate what we've done. And I think actually what's clear to me, I'm a biochemist working, and it's great here with Antonella, clinician, Lucy's a dietitian, and that multidisciplinary approach. And we have our patient organisations over there who are really important to have involved in these debates. So, oh, there we go. Thank you very much.